Hello, everyone, and welcome. It's a pleasure to continue with the Josep Carreras distinguished lectures. And today, this Friday, we have the pleasure to have with us Professor Jan Koels. Jan obtained his PhD degree from KU Leuven in Belgium, in the Flemish Belgium, with the study of chromosomal defects in leukemia. Later, he moved to do his research in genetic causes of leukemia at Harvard Medical School in Boston, where he studied myeloid leukemias, identify a particular fusion translocation, fusion protein. And this fusion protein was responsible of the chronic eosinophilic leukemia. After he went back to Belgium, he became assistant professor, later full professor, and now he's a group leader of the VIB in Flanders. His research is devoted to study genetic complexity in acute lymphoblastic leukemia, mostly TLL, but also BLL. And he's using for this aim single-cell DNA and RNA approaches. He is also a board member of the European Hematology Association. He was editor-in-chief of the open access journal Hematologica, and now he's the editor-in-chief of the new journal Hemasphere that he just told me that is going to have a very nice impact factor. So stay attentive to this, because I think it can be interesting. That it's really a pleasure. The title of his talk is going to be Oncogene Cooperation in Tcell Acute Lymphoblastic Leukemia. Thank you very much.
Thank you very much for this invitation to be here in this beautiful center and to give a talk about our work. It's really a pleasure to be here, and thank you for joining for this seminar. I have no real disclosures except that I'm the editor-in-chief of Hemasphere. I also want to take the opportunity to highlight this to you, and as this open access journal in hematology with the low publication costs, because we really want to support researchers and do not charge thousands of euros for a publication, provide fast review and with only typically one revision. So yeah, consider to publish there. Acute lymphoblastic leukemia is the most common pediatric cancer with a peak incidence in ages 2 to 5 years of age. Most of them are B-cell ALL, and only 15% of these cases are T-cell ALL. And I chose to work mainly on the T-cell ALL. But as you will see, there's also some data on B-cell ALL. The treatment is intensive multi-agent chemotherapy for 2 years in these young children. And the good news is that the 5-year overall survival is high, is more than 90%. T-cell ALL may be slightly lower still than B-ALL. And we hope that by getting more genomic insight, we can further improve the treatment, and further improving treatment can be by identifying these residual leukemia cells that cause relapse in the patients, because the major challenge in these patients and the reason why not all the patients survive over a long time is relapse, and relapse comes from these residual leukemia cells that we do not seem to eradicate with the current chemotherapy. But we also need this biological insight to identify new targets for therapy. Now, this slide shows the typical mutations that we find in T-cell ALL. We've done quite some years ago some exome sequencing, and many other groups like Charles Mulligan has done a lot of sequencing in TLL. And with all these data together, we come to the insight that more or less a combination of about 10 different mutations is causing T-cell acute lymphoblastic leukemia. All the patients have Notch pathway mutations. As you can see here, is a very important gene, Notch or FBXW7. More than 70% of TLL cases have such Notch mutations. Almost all of them have cell cycle mutations, mainly P16 deletion. And each of them also has ectopic expression of a transcription factor, and it's one of these listed here. Some cases have TLX1 overexpression, others have TLX3, others have HOXA, so it's a mutually exclusive overexpression of one of these transcription factors, typically caused by chromosomal rearrangements. And we use this also to subdivide TLL in these different subtypes. We talk about the TLX1 positive cases or TLX3 positive cases. And on top of that, these cases also have then a variety of additional mutations that you see here on the lower or the right-hand side of the slide. Signaling mutations typically can be ABL fusions or ABL mutations, RAS mutations, FLT3 mutations, JAK-STAT pathway mutations, PI3 kinase AKT pathway mutations, and so on. So there's a lot of mutations identified in these signaling proteins, epigenetic proteins, chromatin modifiers. Even ribosomal proteins can be mutated in TALL, indicating that also translation, protein translation can be playing a role in TALL development and some other pathways. It's not surprising that these specific pathways are mutated in TALL because also in normal T cell development, Notch signaling is very important. Interleukin-7 receptor JAK-STAT signaling is very important in initial stages of T cell development, and later on also Hedgehog signaling. So it's logic almost that we find mutations in these pathways because the cells, the leukemia cells, then become independent of ligands that normally will activate Notch pathway, or for example, become independent of interleukin-7, which normally activates the interleukin-7 receptor and the JAK-STAT pathway. So we can see that these signaling mutations most likely contribute and give the characteristic of the cells to become and proliferate and survive independent of the growth factors. Now, when we take a close look at all the different mutations we see in T-cell ALL, we also can distinguish that there's very specific associations between these mutations. The TLX1 positive cases, here highlighted in yellow, typically have NUP214A fusion or PTPN2 deletion or RAS mutations, but rarely have AKT pathway mutations. AKT pathway mutations we clearly see in the Tau1 positive subgroup. So, sorry, it looks like Tau1 requires P10 AKT pathway activation either through P10 deletion or mutations in PI3 kinase units or AKT. OXA is then typically associated with JAK3 mutations, and TLX3 is very specifically associated with FLT3 mutations. So we see these very specific associations, and of course then we start to think, why is that? Is it a genetic cause or is it a biological cause? And so I'll show you some data that shows that there's definitely a biological cause that explains why these mutations occur together. So in the first study, when we looked at TLX1 and the NUP214-ABL1 fusion, we found that when we just express NUP214-ABL kinase in the bone marrow cells of mice, nothing really happens. These mice just age normally, and NUP214-ABL by itself is not a very strong oncogene. Overexpression of TLX1 was shown by the group of Adolfo Ferrando in New York, causes T-cell leukemia, but with a relatively long latency. And when we then crossed the mice with NUP-ABL expression and TLX1 expression, then we observed that these animals do develop leukemia much faster and also with 100% penetrance. So indicating already here that leukemia development is indeed enhanced by the combination of these two oncogenes. When we look to the gene expression profiles of these leukemia cells, and of these mouse leukemia cells, we found that in the mice expressing NUP-ABL and TLX1 together, we had upregulation of a lot of JAK-STAT pathway genes. And all of these genes that were upregulated also had STAT5 binding sites in their promoters. So indicating that it is actually through STAT5 binding that this happens. Here are two examples. MYC expression, for example, is much higher in the NUP-ABL TLX1 animals than when we only express NUP-ABL or only express TLX1. And the same is true for BCL2. BCL2 expression is strongly enhanced if we combine the two oncogenes in the blood cells versus the expression of the individual oncogenes. And we believe that this is most likely going through the STAT5, as we found these STAT5 binding sites in the promoters of these genes that are upregulated. So STAT5 is normally activated in T cells downstream of the interleukin-7 receptor JAK1, JAK3 pathway. STAT5 is a transcription factor, it gets phosphorylated by the JAK kinases and then moves to the nucleus where it binds to promoters and enhancers to regulate the STAT5 target genes. And what we found is that actually TLX1 is a transcription factor and also binds in these same regions. And if we then also express NUP214-ABL kinase, that's a kinase that can also activate STAT5 phosphorylation and STAT5 activation, we can explain in this way that the combination of the expression of the kinase that activates STAT5 and TLX1 that binds the same regions in the DNA as STAT5 can lead to upregulation of these target genes. And so we believe that TLX1 is really potentially not binding its own target sites, but just associates in a way we still don't understand with STAT5 binding here. So potentially to interaction with STAT5 or interaction with other transcriptional regulators in these regions. We use chip sequencing to really confirm that TLX1 is bound together with STAT5. If we look at the BCL2 enhancer here, that's the BCL2 enhancer is in the middle of the intron of BCL2. Here is the promoter, here is the last exon. We see that the BCL2 is active. We have this H3K4 trimethylation mark on the promoter of BCL2, showing that this gene is strongly active in these leukemia cells. And we see that there are 3 nice peaks of TLX1 binding and the same peaks of STAT5 binding, showing that TLX1 and STAT5 bind together here at this enhancer, where also P300, BRD4, and of course H3K27 acetylation is present. When we look throughout the genome, we see that TLX1 and STAT5 co-bind a lot of different sites here. We have 2,500 genomic sites that we look at, and everywhere where TLX1 is bound on these 2,500 regions in the genome, we also find binding of STAT5, binding of P300, BRD4, and we see the H3K27 acetylation, indicating that these are active sites of active transcription. When we then look to these chip data and combine that with the gene expression data, we can distinguish 2 different regions. We can distinguish regions where STAT5 is bound and where STAT5 is causing upregulation of those genes. And on the right-hand side of the slide, you see regions that are bound by STAT5 also, but where the transcription is downregulated. So STAT5 can both up or downregulate certain genes. And when we split it in these 2 types of genes, we can see that where STAT5 is binding and upregulates the genes, we find MYC and MAX motifs also present, indicating or strongly suggesting that MYC is also binding these regions together with STAT. And where STAT5 is more downregulating, we see more ETS factors that are bound there. So when we combine all that, we indeed see that in those regions in the leukemias where TLX1 is bound and STAT5 is bound, also MYC is bound, ETS1 is bound, and RUNX1 are bound. So it's a big hub of many different transcription factors. And TLX1 is probably just tilting that balance of those regions. These regions are normally bound by STAT5, and when TLX1 is overexpressed now in the leukemia cells, then TLX1 enhances the transcription from these sites. So we conclude from these data that what happens in the leukemia cells is that we have TLX1 that is co-binding with STAT5, and the NUP-able kinase is just activating more STAT5, so that these regions, enhancers or promoter sites where STAT5 is already normally binding, is now upregulated even more, and we get higher expression of MYC. MYC is a very important oncogene in T-cell ALL. We get higher expression of BCL2 as an important anti-apoptosis factor, and so on. And this drives these leukemia development and the cooperation between these 2 oncogenes. So this explains nicely why TLX1 and NUP-able can work together. And I just want to show you a few additional examples where we see HOXA9 and JAK3 cooperation, also TAL1 and AKT. And in each of them, it's slightly different. It's not always exactly the same mechanism that is used by these different oncogenes. In case of JAK3 and HOXA9, it's very similar to the story I just showed with TLX1 and ABL. Here, JAK3 also activates STAT5 phosphorylation, and HOXA9 also binds at the same regions as STAT5, but here more at promoters than at enhancers. And typically, again, in these mouse models, we see that when we express JAK3 mutant and HOXA9, we get leukemia development much faster. Within the first 100 days, the white blood cell count is largely increasing. The cells that really contribute to the leukemia express both JAK3 and HOXA9. And you see that the survival of these animals is also much shorter than HOXA9 or JAK3 alone. Our institute also asks us not to do all the experiments in vivo in mice models and try to reduce mouse models. So we also try to see if we can develop other systems, other assays that do not need live animals anymore. And so we've come up with this Protessa culture that we can establish from a single mouse, contributes bone marrow cells. And if we put these cells in the presence of stem cell factor, interleukin 7, and uncoated DLL4, that's the ligand for Notch, then we can differentiate these cells towards T cells. It's immature T cells that we maintain. These are primary cells. We can generate these from every type of mouse. So you can use a wild type mouse, but also transgenic animals to have here your Protessas with already certain defects. And these cells can be maintained in culture for several weeks to several months. They do not become a cell line and they remain primary cells. And some people in the lab have sometimes more difficulties culturing these cells than others. So there's some tricks that you need to know, or you need to carefully take care of these cells to grow them. But I think it's a nice system that allows us to also look at oncogene effects in these cells ex vivo. Now, in this example, we used Pro-T cells from a CRE-ER mouse, where we can then activate CRE by addition of tamoxifen in these cultures. And in this way, we can use an inducible retroviral vector to induce the expression of certain oncogenes. Here we've done that for Tal1, one of the main transcription factors in T-ALL. And we've used here an inducible expression because if we just express Tal1 in these T cells, then the cells do not like this, and these cells will undergo apoptosis and slowly disappear from the culture. So Tal1, although it's an oncogene, is really not liked by T cells initially. And it's only when we express Tal1 together with AKT pathway activation that we can culture these cells, and that it becomes an advantage for the cells to have both Tal1 and AKT. Here you see in the black solid line what happens if we start expression of Tal1 here in these cultures. You see that these cells with only expressing Tal1 slowly disappear from the culture because the wild type cells not expressing Tal1 grow much better than those expressing Tal1. But if we express Tal1 together with AKT, then this is the dotted line, then we get expansion of the Tal1 AKT cells. And so only that combination seems to be beneficial. And that can explain here, I guess, why Tal1 is always found in combination with AKT pathway activation. And other pathways cannot really help. We also tried with the RAS activation or with JAK pathway activation. That's not good. It's only AKT pathway activation that is good for Tal1 expression. Similarly, we used here then Cas9 mice to derive again the same Pro-T cells, but so now they will also express Cas9, and in this way we can also test what happens if we knock out genes. And we use this to look at the cooperation between mutant JAK1 and inactivation of Sus12. Sus12, one of the proteins of the PRC2 complex. And if we express JAK3 alone or inactivate Sus12 alone, nothing really happens in these cells. They cannot really grow in the absence of interleukin-7. But if we express JAK3 mutant together with a guide RNA targeting Sus12, then we can make these cells interleukin-7 independent, as probably also the leukemia cells, the real leukemia cells become interleukin-7 independent. So I just wanted to use this to show that in these ex vivo cultures are a really nice system to study different aspects of T cell proliferation and survival, potentially also differentiation. So with this, I showed some examples where these genes indeed work together and where we have a combination of ectopic expression of a transcription factor with a signaling mutation that is really strongly driving leukemia development. Now the question arises then, of course, if we look at all these sequencing data, it's nice to see that there are Notch mutations, that there are kinase or RAS mutations. But the question is, do all the cells actually have these mutations? Are all the cells in the leukemia samples the same, or are there differences? And the first observation that we made was already quite some time ago in 2015, when we were looking specifically at the JAK3 mutations in T-ALL. And we found that about 30% of the cases with such a JAK3 mutation have actually more than one JAK3 mutation. So every patient there had just not one, but two, or sometimes even three different JAK3 mutations that we could detect with deep sequencing. So we typically have an M511I mutation. That's the most common mutation in JAK3 that we find. But on top of that, we found that in most of the, or at least in 30% of the patients, we had additional mutations in JAK3 in other domains that we were also finding in the same samples. And initially, we didn't understand that very well. We didn't know what is happening in the same cells or is happening in different cells. And with this bulk sequencing, you can never know whether these mutations are present in exactly the same cells or in different cells. So we need to turn to single cell sequencing to define that. And that's how we started to do single cell sequencing in T-ALL to further refine this genetic information that we have by bulk sequencing. With single cell sequencing, we can then address questions first of all, to see are there different clones, different mutations present in different cells at diagnosis, but also in what order are the mutations potentially acquired, and also how do these different clones evolve during chemotherapy treatment. So we set up a first set of experiments to look into this. So in theory, we thought that most likely we will find different clones. Because if you think about how leukemia is developing, you initially have one cell that acquires a first mutation, and this is the blue cell here, and this cell is not changed by this first mutation a lot, so it behaves quite normal, can proliferate and differentiate. One of these cells with the first mutation then acquires a second mutation, here indicated then as the red cells. And of course, also that is still definitely not sufficient, because when we diagnose patients with T-ALL, we always find almost 10 or more mutations present in their genome. So we need accumulation of additional mutations. And so there's a third mutation probably acquiring, these are the green cells, and a fourth mutation, the yellow cells, or potentially at this stage, it can also start to diverge, and you can have different sets of mutations. So here the yellow and the purple cells have the same 3 mutations in common, but then have different mutations at the end. And so when we think about it, just in theory about that, that's potentially what we can identify in a leukemia patient at diagnosis. You should be able to find all these different cells back at diagnosis. Also, still the cells with only 1 mutation or with the first 2 mutations. And in this way, you can then predict what is the order of the mutations that these mutations were acquired in the patient. So initially we started with a very basic way to look at single cells. We sorted 384 cells in 384 well plates, extracted DNA, amplified DNA from these cells, and then did PCR and sequencing to identify which mutations were present in the cells. And already at that stage, with this simple method, we only did 4 patients at that time. We found that there was here in this patient a set, about 25% of the cells that did not have the notch mutation yet. 66% of the cells had all these mutations plus also a notch mutation, and there were 25% of the cells in which we could not detect the notch mutations, indicating that indeed with these single cell methods, we can identify subclones of cells with only the first mutations, and even here we had a few cells with only the first 3 or 4 mutations and nothing else. But that was still only in 384 cells, so that does not allow to really make very strong conclusions. So we needed to look for a better method. And then we started some years ago to work with the Mission Bio Tapestry platform that allows you to generate custom panels to do amplicon sequencing. So we had 305 amplicons designed for all the most common mutated regions in ALL. So we can, in the single cells that we generate with this method, do these amplifications, PCR amplifications, in every single cell, 305 PCR amplifications, and then sequence to detect the variations and also small indels. The way this method works is by droplet generation. So we have the cells that will get into an oil so that every droplet has only 1 cell. You can also add proteins if you want. So antibodies detecting cell surface markers, but we did not use that initially. And in each droplet where there is 1 cell, then there is a second droplet fusing in, which contains the beads and the reagents to do all the PCR. So within such a droplet, the cells is finally then lysed, the DNA is released, there's PCR amplification of these 305 regions, and each of these PCR fragments also has a unique barcode that tells you that this is the cell where this PCR fragment is coming from. So for each of these cells, we get information on these 305 amplicons. We generated a custom panel with the most commonly mutated regions for B and T ALL initially. We had a coverage of about 110 genes and more than 1,300 reported mutations. And since then, we've now designed a new panel, specifically one specific for B and one specific for T ALL. But the data we'll show you today is coming from this custom panel for both B and T ALL mutations. First of all, if we look at the panel performance, when we group all the data together that we got from the different samples, we found that there's definitely a few of these amplicons that do not perform well. So we lose information on these amplicons because they're not covered well enough in each of the cells that we analyze. And there's also a few amplicons that are actually covered much too high, 600 times instead of about 100 on average. So we have much more information from some amplicons and not so good from others. But in general, I think we were satisfied. About 95% of the amplicons had at least 10 reads per cell, and that's sufficient to do our analysis. Typical cell numbers we get in these methods is in the line of about 4,000 to 5,000 cells, but also lately we are improving that to 6,000, 7,000 or 8,000 cells per sample. The coverage is about 100x on average, but variable, of course. And the allelic dropout that we had was below 10%, which is acceptable, but it's still something to take into account. Allele dropout is a problem in single cell analysis. It means that you think your cell is homozygous for a certain mutation, but it could have been heterozygous because just due to the error of measuring, we cannot always get the information from both alleles, but that's to a limited number. So then when we first look at the T-cell ALL, so we covered more or less these different genes, and notch mutations are definitely of interest because 70% of the cases have notch mutations. And then of course also signaling mutations are interesting to look at. So for notch, it's really surprising to see that in almost each of the T-ALL cases that we analyzed, we always find more than one notch mutation. So the way we read these samples is start on top. So we have 3% of the cells were still wild type, so have no mutations. But then when we go down, we see that there is about 10% of the cells that have these mutations here, a RAS G12D and a KLF mutation. And then these start to split up. Some cells have this notch mutation, and other cells will have another notch mutation to arrive with clone 3. And clone 3 can again be split up. Some cells have a second notch mutation on top, and others do not have that mutation, but have an IKZF1 mutation. So you see there's a lot of different cell types that we can distinguish. Even here, 2% of the cells do not have notch mutations at all, but just have a different RAS mutation. Most of them have the RAS G12D mutation. These have a RAS G12C mutation. So indicating that these leukemia cells apparently choose sometimes different ways to end up with different types of mutations. And so these are all diagnostic samples. So much better than at the bulk sequencing, when we performed whole genome sequencing, we had only identified the RAS G12D mutation and this one notch mutation, but actually the patient has 2 different RAS mutations and 3 different notch mutations. So the single cell sequencing can really add additional information based on top of what we can see with bulk sequencing. And we also see that in the other patients. We see different notch mutations. We see different notch mutations also here in this patient, 2 major clones with 2 different notch mutations, and here also again 3 different notch mutations. And some cells end up with these 2 notch mutations, while other cells in this patient have only 1 other notch mutation. And so that indicates that the cells somehow independently from each other can acquire notch mutations at different times in the evolution of this leukemia. It indicates that notch mutation is really important for these cells because they acquire it independent from each other, but it also makes it complex for molecular diagnosis because which one is now the most important notch mutation in these patients? Probably all of them are important. And a similar thing we find when we look at the signaling mutations. Signaling mutations here, JAK3 mutations or JAK1 mutations, you see that this patient also has variant roots. Some cells, only 1% of the cells has here this JAK mutation, while the majority of the cells have another JAK1 mutation on top of a JAK3 mutation that was acquired earlier on. So also in these signaling mutations, we see that there's a variety of cells. Some cells have a JAK mutation, other cells do not have a JAK mutation. And we see that in more over and over again in the different patients that we analyze. Then we also looked during treatment what happened, and first of all we checked whether bone marrow samples and blood samples had more or less a similar heterogeneity and similar clones present, and that's indeed the case. When we look to each dot here is a different combination of mutations, so we find that back in bone marrow and also in blood at more or less similar percentages in most of the patients. There is a few exceptions, but most of the mutations we're looking at and clones we're looking at are equally represented in bone marrow and blood, because also during treatment from some time points we only have blood samples, and for other time points we have bone marrow and blood samples, but we believe that we can use these equally. So this is what happens in a patient that was treated with chemotherapy and that we followed over time. Initially, we were able to identify a WT1 mutation in some of the cells, only a WT1 mutation. Some cells had a WT1 mutation plus 2 different notch mutations, and some cells had a WT1 mutation plus only 1 of the notch mutations, and also some wild type cells, of course. And so during treatment, after 5 days of treatment, you see that the number of cells with mutations is already decreasing. After 11 days of treatment, most of the leukemia cells have disappeared from the blood, and we only detect cells without mutations. And after 35 days, we have a bone marrow sample and a peripheral blood sample that we analyze, and we can here still detect about 45 cells, especially from this clone here, the dark blue clone, with only the WT1 mutation. So indicating that this clone is a little bit is remaining longer, and the other clones seem to be more sensitive to the treatment and have disappeared faster. In the second patient that I show you, we looked at relapse in this case. So we had a diagnosis, a major Notch1 clone with this Notch1 mutation, and also some cells without mutation. And 2 years after the initial treatment, these patients relapsed. And when we then analyzed the bone marrow and a peripheral blood sample, we identified a totally different Notch1 mutation to be present in the major clone, and some minor clones with additional different notch and FBXW7 mutations. This patient relapsed for a second time, 4 months after the first relapse, and then had again acquired a totally different set of mutations, a P10 mutation, P10 plus JAK3 or P10 and P53 mutations. It was interesting to see in this case that when we look back then to the diagnostic sample, we had indeed 4 cells here at time point 0, so 2 years before the relapse, 4 cells that had already this notch mutation that was the major clone here at relapse, and all the cells had a different notch mutation. So we could actually detect already the relapse clone here at diagnosis, but it was a very small clone at that time. And looking back, these were the cells that somehow were resistant and were causing the relapse later on. So in conclusion, I would say that in this single cell sequencing identifies clearly these many different subclones in the majority of T-ALL cases. We see various notch mutations, various JAK-STAT and RAS pathway mutations as the major drivers of this heterogeneity in T-ALL. We can identify residual cells during treatment, and we can also go back and see these relapse clones at diagnosis. I want to switch very briefly to B-cell ALL to also show how it's in B-cell, and there it's slightly different than in T-cell ALL. Now we have, of course, a different set of mutations that we need to look at, but the signaling mutations are very similar between B and T-ALL, but we also have more and additional transcription factors that are mutated in B-cell ALL. We analyzed 12 B-ALL cases with the same methods, again with bone marrow and blood samples, and also some during treatment. But the major message I want to give is that in B-cell ALL, this heterogeneity is clearly less pronounced than in T-cell ALL. We have some cases here with EDV6 RUNX1 fusion or BCR-ABL fusion, BCR-ABL-like, or with this IKZF1 fusion, mutation. All these samples did not show a lot of heterogeneity, and we almost did not detect any additional mutations on top of the oncogenes that are shown here. But in the cases with the PAX5 alterations or in the high hyperdiploid cases, there we do find a lot of additional mutations, and we also find heterogeneity in the samples. For example, these 2 high hyperdiploid cases here, we find that there is an amazing amount of different signaling mutations. And some cells have a FLT3 mutation, and so cells are shown here in columns. So every column is a different cell. So some cells have a FLT3 mutation. Other cells, very minor population, have this RAS G12D mutation. Other cells have a PTPN11. Other cells have another RAS mutation or even a BRAF mutation. So a quite variety of mutations, all similar signaling pathway mutations activating the RAS-MAP kinase pathway. FLT3 is also activating the RAS-MAP kinase pathway. But different cells, again, have different mutations. And also in the second patient here, the same story, where we see some cells with a FLT3 mutation, some cells with KRAS mutation or NRAS mutations, a lot of different mutations present in the same patient. And for FLT3, it was even more surprising to see that we have a whole variety of FLT3 deletions. Deletions here in the juxtamembrane region that are all in-frame deletions, so they're resulting in the activation of FLT3. But all these different deletions that you find here were coming from the same patient. So from different cells in the same patient had different types of FLT3 indels, indicating again that this all happened independent from each other during the leukemia development process, indicating that it's really needed for these cells to acquire FLT3 mutations, but also indicating that it's very difficult when we do molecular diagnosis if we have to deal with so many different mutations present in the different cells. So in conclusion, also in B-ALL, we see heterogeneity, but it's less pronounced than in T-cell ALL. We see it mainly in the PAX5 alterating cases or in the high hyperdiploid subtypes, and others might not have such a heterogeneity. But that brings us to some important consequences for molecular diagnosis, because typically we report that the case has a RAS mutation or a certain FLT3 mutation, but here we see that there's often much more than one mutation present in these cases. And I believe this also has important consequences for targeted therapy, because if we start to treat such patients with a FLT3 inhibitor, but there are also cells without a FLT3 mutation and with a RAS mutation, that will cause very rapidly resistance to the treatment. So we have to be cautious with that, and we need to know this heterogeneity. Now we can think of, just also look at the time. Okay, we can think of targeted therapy in these cases, in T-cell ALL or B-cell ALL. There's not that much targeted therapy used at the moment, but hopefully in the future, we can bring in more targeted therapy to treat these patients. And we can think typically of targeted therapy based on genetics. If there is a FLT3 mutation, we would treat with a FLT3 inhibitor, or if there is a JAK1 mutation, we would treat with a JAK inhibitor. But as I just showed you, many of these cells have in the same patient different types of mutations, and it's likely that such approach will probably not work and that resistance will develop very rapidly. So we probably need to look at the mutations that are present in almost all of the cells, and these are the transcription factors that are expressed. We believe that these are very early mutations, and so that these mutations could be targeted, but it's also a class of mutations that is difficult to target at this stage. We could think of the cell cycle or notch pathways, which are more common and which are present in more cells, but still this brings us to some problems also with notch mutations. We clearly see that not all the cells have a notch mutation in such a leukemia patient. So again, there is also some problem. So I would strongly suggest to not look too much into these mutations when we choose to go for targeted therapy in T-ALL, and that we look more to general pathways that are based on biology that we know are very important to most of the cells or all the cells in the leukemia sample, independent of the presence of certain mutations. If we want to target transcriptional regulation, then BET inhibitors or CDK7 inhibitors might be a choice in the future. If you want to target apoptosis, there's a number of inhibitors, a number of studies showing that indeed we can induce apoptosis with BCL2 inhibitors. And there's three pathways or three classes that I show here in red: the notch pathway, the glucocorticoid receptor, and the nuclear export that I think are most interesting to target in T-cell ALL. Notch pathway, because most of the cases have a notch mutation, and we have gamma secretase inhibitors available to target notch pathway. The glucocorticoid receptor is already used. That can be activated with dexamethasone or prednisolone, and the glucocorticoid receptor activation will result in induction of apoptosis. So that's one of the cornerstones of the current treatment, and we can probably improve on that. And also nuclear export seems to be a pathway that is very important in these acute lymphoblastic leukemia cells, both T and B ALL, and which we can target with XPO1 inhibitors. And XPO1 is a protein that regulates the nuclear export from a number of proteins from the nucleus to the cytosol. And for some reason that nobody still understands, leukemia cells are more sensitive to inhibition of XPO1 than wild type cells. And so that's definitely also one way to go. It's a very common protein in all the leukemia cells, and it's independent of mutations. So we've done a lot of studies with XPO1 inhibitors, and we have not seen any differences between the different types of B-ALL or T-ALL cases. All seem to be relatively well sensitive. So let me end by just showing a few of those data and how we could approach this to treat T-cell ALL. The first one is gamma secretase inhibitors. Gamma secretase inhibitors have been around for a while, and there's not much belief anymore at this moment in gamma secretase inhibitors because most of them have shown a very strong gastrointestinal toxicity. But we found that you can also make selective gamma secretase inhibitors. So selective meaning that you don't target all the gamma secretase complexes in the cell, and that you selectively target a few of those complexes, because the gamma secretase complex is actually consisting of many different proteins, and not every complex has exactly the same proteins present. So you can selectively target some of these gamma secretase complexes. And with, for example, this inhibitor that we identified is one of these selective inhibitors, MERK560. It potently inhibits the growth of these leukemia cells. This is human leukemia cells in an NSG mouse. It potently inhibits the growth of these cells. So this is only treatment for 2 weeks with this MERK560 compound. Of course, it will not cure these animals, but it clearly has a strong effect already. And it is not toxic at all. MERK560 does not cause any gastrointestinal toxicity. It's the same as vehicle, while the other previously used gamma secretase inhibitors cause this goblet hyperplasia that causes gastrointestinal toxicity. So if we look into more specific inactivation of these complexes, that can be one way forward. Second data is on the XPO1 inhibition, so the export inhibitor. There, KPT-8602 is one of these inhibitors. By itself, it has activity. You can decrease the leukemia in these animals. Again, this is the human leukemia cells in an NSG mouse. You can inhibit partially the growth of these leukemia cells in the mouse. But if you combine it with dexamethasone, then you get a much stronger effect. So the combination with KPT and dexamethasone is very strong. And as you can see here, especially in the bone marrow, you will see that dexamethasone by itself or KPT by itself does not really target the leukemia cells in the bone marrow compartment. But when you combine the two drugs, then you really can also have a strong effect in the bone marrow. And then finally, we are now trying to combine all these inhibitors together, where we inhibit the gamma secretase complex. That's the darker blue curve, where we get already some effect. If we activate the glucocorticoid receptor with dexamethasone, we get a similar effect. If we combine these two drugs, we get more effect, longer survival of the animals. And if we add also the XPO1 inhibitor, then this experiment is still ongoing, and we could almost completely eradicate the leukemia cells with this triple combination of drugs. And so this is just targeting very general pathways in the leukemia cells, not really looking at certain types of mutations, and that, of course, potentially combined then with chemotherapy in the future could, I guess, be a way forward to further improve treatment. So in conclusion, I would say that we have some good ideas now based on all the mutations present in TLL, how they work together, why certain combinations of mutations are found in T-cell ALL and how they cooperate. Based on single cell sequence data, we get new insight in the heterogeneity of a diagnosis, the different clones and different mutations that are present at diagnosis, and especially in this a lot of different signaling and notch mutations that are present in TLL in different cells at diagnosis. And that brings me to the conclusion that it's probably not so good to target signaling mutations in ALL because it's just too much heterogeneity at diagnosis, and cells will easily escape from that type of therapy. And it is probably a better way forward to look into general proteins that are present in all the cells, such as exportin or gamma secretase complexes or other types of proteins that, for example, are involved in transcriptional regulation or translational regulation of proteins. And that's also an advantage because we don't need to know the mutation status of these cases, which is typically taking a long time and can also give us maybe the wrong information. With this, I want to end and thank the members of my group here. Not all of them are on the picture, but you see the names there. We also work closely together with the hospital. Our campus, research campus, is just next to the hospital in Leuven, so that allows us to get samples from the hospital in an easy way and a fast way, and also for all these single cell analysis, that was definitely important to freeze the samples in the best possible way. And we definitely also work together with other members of our university. And at VIB, we have a tech watch team that helped us a lot to scout for new technologies that are available to use in our studies. And with this, I thank you, and I thank you for your presence.
Thank you very much. Was great. Thank you very much. Was great. I like it very much. I have before giving opportunity to the others, just very simple questions. In your single cell, in that case, that relapse, at the end you have P10, P53, P1053. Some of these were observed also at diagnosis or not?
No.
Not
Not
even one cell.
Not even one cell. Of course, we only analyzed about 5,000 cells, so we could have missed it, but we did not detect it.
But do you think that they were there or not?
Probably not.
Probably not.
That would be my guess.
Yes.
From the first relapse, we detect some cells, and we can estimate that these are the cells from the first relapse. But the second relapse is something that happened very quickly, but we did not detect these cells.
And in the first relapse, the P53 was there or not? In the first relapse?
No.
Not. Okay. Very good. Yes. A second question is, have you analyzed, when you use this combination of drugs now in the last part of your talk, are you analyzing the dynamics of the single cell mutation there? What's happening upon the use of the drugs?
That's currently something we're doing, because also you could expect that maybe some clones are more resistant to others. So we have these PDX samples. So from each of the samples that we have done now, the single cell sequencing, we still have frozen samples available that we can inject in NSG mice and then treat with different drugs and look at these dynamics. Yeah, that's a very good point, and that's something that will take some time to do, but that is upcoming. Yes.
Thank you very much. Questions?
Hi, thanks a lot. I'm not sure if you have this data, but I always wonder if you take biopsies from different parts of the bone marrow and would do this analysis of these beautiful trees of mutation, how much difference would you observe? Particularly taking into account that in Spain, we take the biopsy from a different point than in the Northern European countries. Yeah,
that's a good question. And I do think that most likely you could find some differences. That can also be the reason, for example, why we missed the P53 mutation, that is just somewhere else and that we only have a sample from one space. So I think that's a good point. It's of course, it's a difficult ethical thing to take bone marrow samples from different locations. I think that I'm not sure if the ethical committee would allow us to do that, to get a view on that. But at least the only thing that we can compare is that bone marrow and blood seems to be very concordant. So that's what we see in the blood samples is also present in what we see in the bone marrow samples, and we have not detected major differences between bone marrow and blood, which seems to indicate that at least if there are differences, they're going to be minor, probably. But I totally agree. Yeah, different sites, different bones even can harvest different clones. Yeah.
Thanks.
Beautiful talk, thank you. So I have two questions. One is regarding your first part on the NUP-able cooperation with Tcell to activate type 5. So you think that JAK-inhibitors would interfere with that upregulation or activation of that 5 by shifting that balance, and that could somehow reduce the leukemia development?
Mm-hmm. Yeah, so we have used for the NUP-able, we have able inhibitors like dasatinib, for example, that can be used, and we've used that. And of course, in the mouse model that works, because in the mouse model, we probably don't have that heterogeneity. Also, we just induce expression of able and we use expression of TLX1, and then we get this leukemia, all the cells are the same. There it works. But I think in patient samples, we did not analyze a NUP-able positive patient sample. It's quite rare. So we didn't have that in our collection. But I think it would resemble probably very similar to what we see with the JAK and the RAS mutations present in the same patients, or different JAK mutations present. So I would guess in a patient, it would be more difficult to achieve such a treatment. I think it's totally different than BCR-ABL. BCR-ABL is a driving oncogene, which probably happens much faster, and all the leukemia cells will have BCR-ABL. But in these NUP-able or JAK mutations are happening so late in the leukemia development that you have cells with and without such mutations, different mutations, that this variety of mutations is too big, at least in some samples and maybe not in all, and that there the kinase inhibitor would have limited effect. I think in AML, there's been a nice study with single cell sequencing where they treated patients with AML and FLT3 mutations with a FLT3 inhibitor, and they saw that the RAS mutations were coming up very fast because they were already present at diagnosis in a minority of cells, and that caused then relapse in these patients. So I think similar things we can expect in ALL as well.
Okay, and just very quickly, the ex vivo model that you showed, where you put the SCF, IL-7, did you try that with human samples? Could you have that model in humans? Because
we did not use human samples, but that's a good point. And there are labs who use that model and who develop that model with human cells. So that would also work. If you start from CD34 positive human cord blood cells, for example, you can also have them differentiate towards immature T cells in these ways.
Thank you.
Thank you. That was very nice, very inspiring. I wanted to ask you about the gamma secretase inhibitors because in a way, the way that gamma secretase inhibitors work, if they inhibit Notch, they should inhibit also the intestinal role of Notch in intestinal development. And I was wondering, do you have any idea why this inhibitor doesn't work intestinal toxicity?
We have a hypothesis. It's not completely confirmed yet, I think. But so what we see is that the gamma secretase complex, an important protein in the complex is presenilin, and presenilin 1 or 2 can be expressed in cells. And so some complexes will be presenilin 1 containing complexes, and other will be presenilin 2. It's 2 different genes. We see that in the leukemia cells, only presenilin 1 is expressed, not presenilin 2. So if we inhibit presenilin 1, there is no other presenilin anymore. So the cells do not have any gamma secretase complex at all anymore. While in the gut cells, we see that both presenilin 1 and 2 are expressed. So if we specifically inhibit 1 with the drug, then 2 is still there. And apparently that could potentially take over or will be sufficient to have sufficient gamma secretase activity left. It's a hypothesis. We don't see any toxicity with the drugs, and we compare that with a general inhibitor. So most of the other gamma secretase inhibitors inhibit both presenilin 1 and 2 complexes. So that's why you always have a complete inactivation of the complex. But with the more selective drugs, you could potentially rescue the normal cells by just inhibiting one of the 2.
Okay.
But we still would need to do more experiments to really confirm if that's the case.
My other question is only some of the Notch mutant cells, the ones that have the mutations in the heterodimeric domain, would respond to gamma secretase inhibitors, but not the ones that have it in the degradation domain. So are you distinguishing any, so like, for example, the patients that you have analyzed, or the xenografts that you have treated, were they?
Yeah, they were all responsive because at the end, Notch needs to be cleaved anyway, independent on where the mutation is, except of course if you would have a chromosomal translocation leading to expression of the internal part, but that's very rare in leukemia. Most of the patients have a mutation, and no matter where the mutation is, it always needs to be cleaved by gamma secretase. So it would also have an effect. We also have even a sample with an FBXW7 mutation, which is the negative regulator of Notch. That one also responded to the gamma secretase inhibitor, because also there you still need cleavage of Notch. So I think it's most of the samples should respond, and that's also what we see. We have analyzed many different samples with many different mutations. But again, if not all the cells have a Notch mutation, then that can still cause a problem. So the fact that we see that Notch mutations are relatively late in the development of the leukemia is worrying, I find. I'm not sure if the gamma secretase inhibitor at the end will be the best choice for treatment.
Hi, thank you for the talk. You are mainly concluding on leukemia in general, but how similar or different are myeloid and lymphoblastic leukemia in terms of genetic mutations, and how a comparison between the two could be useful in terms of drug combination and drug development.
Yes, so also in AML, there is quite a lot of heterogeneity observed. We have not done any studies there, but there are other groups who have done sequencing in similar ways on AML samples. Of course, not every sample has that, but they're also on signaling mutations, apparently, that is RAS mutations, FLT3 mutations, JAK mutations that also occur in AML. We also see this heterogeneity there, that there are multiple mutations present at the diagnosis. And that links well with the fact that it's probably more or less the development of ALL and AML is in that way similar, that there are first driving mutations, which are the chromosomal translocations or the MLL fusions, other transcription factor fusions that are generated in AML. That's probably similar to ALL. And only very late are these kinase mutations and signaling mutations acquired to really drive then the progression and the final acute phase of the leukemia. So that's why probably there also they are not so important. In chronic myeloid leukemia, that's then a different type of myeloid leukemia. There, BCR-ABL, for example, is the driving force of the leukemia. So there, I think it's you see much less heterogeneity, and there BCR-ABL is one of the first mutations, or maybe the only mutation acquired in CML. And that's a difference. So it's not always, it's not just that the kinase mutation is always late, it's dependent on the disease, that's true. But I think in ALL and AML, that is both going to be the case. Some of these drugs that are used in ALL can also be used in AML, like the exportin inhibitor is also, seems to be also active in AML. The first exportin inhibitor is also a bit toxic, and that's a problem, of course, also because it's good, but it's also toxic. It's not better than chemotherapy in that way. There are second generations of these exportin inhibitors that are now being developed, that the one that we use, that seem to be less toxic, and that can have, in this way, maybe a place in future treatment, but that still needs to be looked at, of course. And I think also the inhibitors of transcriptional regulation can potentially both be used in myeloid and lymphoid acute leukemias. They probably also have similar effects there. But yeah, they're also, of course, they also work on normal cells. So that's always finding that therapeutic window between the leukemia cells and the normal cells to really see what can work. But I think in general, AML and ALL seem quite similar to me.
Okay, but in that sense, drug combination is a good way to reduce some clones development to target most of the cells.
Yes, and I think where these newer drugs can then have a place is in ALL and also in AML. So MRD is important if a patient has too high level of residual disease still left after 30 days of treatment or longer days of treatment. That's definitely a problem, and we know that's a high risk for relapse then. And typically in ALL, these patients could potentially go to transplant if that's possible. But here maybe also by using other drugs than the chemotherapy that was used, maybe you can target these residual cells. Again, I doubt if some of these kinase inhibitors will have a role there, but maybe the more general inhibitors like exportin inhibitors or transcriptional regulator inhibitors could play a role there to further reduce these residual cells.
Thank you. Today, in the interest of time, we'll finish just with one question, because this is done in a virtual manner. So we have a remote audience asking around, and it was related to this, to the toxicity. When you increase the number of drugs that you give to your animal models, to your living models, do you see an increase of toxicity, or they remain the same?
We do measure toxicity carefully also in the animal model. So in the last model I showed you where we combined the 3 drugs, yes, we do see a little bit of more toxicity, definitely. And so mainly as weight loss of the animals, and that's, but it's not going to the level that we should stop the treatment. We have a cut of 10% weight loss is what we consider as when we should stop treating the animals. So in that way, it's acceptable, but it's definitely a consideration. The question, of course, is if you need to use all the drugs together, or if you can use them consecutively, or first a round of chemotherapy and then only use some of these drugs. That might be tolerated better than all these combinations together.
Thank you very much. Congratulate the speaker again. Thank you.
Thank you.